Okay, so first of all, can you hear me from the back? No problem? Good. I don't like to carry microphones around. As you probably realize, this is my first day teaching here. I came here to visit a couple of weeks ago just to see the environment and not to be surprised by what I could find. And what you need to realize is this is a historical moment because it's my first lecture at George Mason ever. So you can now tell your grandchildren thank you. On September 27, I was there. I witnessed something very extraordinary. But I want to really tell you, I don't know what you guys know or not, but I'm here simply because somebody else cannot be here. This course was taught in the past by Dr. Jack Yu, who everybody knows, and Dr. Peter Andrews. And unfortunately, Dr. Peter Andrews passed away about 2 weeks ago, maybe even less than that. He's been sick, and of course they had to find some other individual to come and tell you all these exciting things about all these tissues. And they tapped on my shoulder, and I said yes, and I'm here. So just to prevent any doubts, I am definitely not a histology expert. I am a molecular oncology expert. I do mainly research on cancer, and I've been teaching many other things that from one year to the next, they change. The good thing about histology is that a neuron is a neuron today. It was a neuron 500,000 years ago, and it still will be when I'm not here teaching anymore. So what we're going to be doing today is, the best way to get me into the system is to ask me to do 2 lectures in one lab, just in case I need enough pre-warming. And we're going to be dealing with the nervous tissue. Okay? So there is a long list of learning objectives, but fundamentally what we want to try to understand is the different divisions and components of the nervous system, the different types of cells and the different functions of these cells, and how they coordinate with each other. So the learning objectives are first followed by the appropriate kind of contents for the lectures, and this is exactly what we're going to be dealing with. So the nervous system has 2 main divisions in humans and the majority of mammalian organisms. They have the central nervous system, which is formed by the brain and the spinal cord, and the peripheral nervous system. The brain and the spinal cord are kind of the command posts, which are going to allow the individual to interact directly, communicate, and react to changes in the environment. And the environment I want to qualify is not only the environment outside. It's not the environmental outside of the body. It's not these problems with the climate change and all these kinds of things. It's also the intrinsic environment. We have an environment inside us that could change by different physiological conditions. And the nervous system is also in charge of taking care of realizing that everything is the way it should be and that we are in the best conditions to perform physiological normal functions. So the main command is the CNS, and the peripheral nervous system is what really gets the CNS, the central nervous system, in control and in touch with everything else. The functions of the nerve cycle, of the nerve cell, had basically 3. The irritability, which is simply the ability to respond to a stimulus. Conductivity, and that's a key function of the main type of cell that is typical of the nervous system, which is the neuron, is able to produce kind of an electric current that is going to perform the function of transmitting the signal that's received from the outside or the inside to the proper place and elicit a response. And then there is a secretory function, because most of the transferring of the signal that's received from somewhere is relayed to the next kind of neuron by secreting certain type of what is called neurotransmitters at different points in what is called the synapse. We'll look at all these things later. The main type of cells that we will find in the nervous system are the neuron, which is a prototype, the glial cells or glial cells, depending on how you hear how to pronounce it, they are kind of helpers. They have primarily a function of protection. And we'll deal with another set of mechanisms of protecting the nervous system in the second lecture. And then almost everything is kept together by connective tissue that is going to be involved also in the maintaining the proper functioning of the work. So this is kind of the general picture. It's actually taken from a book that I was told that you guys have been recommended. And you can see that is the brain as the center of the whole story. This is the spinal cord, and then a number of connections with just about everything, which is done through the peripheral nervous system. And there is two kinds of mechanisms in terms of the way the signal is sent from one part to another. The afferent neurons are the ones that are going to be receiving and transmitting to the central nervous system. And then once the signal is received by the central nervous system, it's going to send something to wherever it has to go. And these are neurons which are particularly adept in sending these responses, and these are called the efferent neurons. So afferent, efferent, if you guys have any question or any problem, remember which one is which. Remember that exit starts with E, and that's the same way that efferent are. So the exiting signal from the brain to the central nervous system is going to be the efferent type of neurons. In terms of types of neurons, we can classify them, and remember that scientists like to call things names because they need to understand each other, particularly morphologists and anatomists and histologists like to do those kinds of things. And thank God they don't do Latin and Greek anymore, although many names are based on Latin and Greek. So depending on the location of the so-called processes, the neurons have what we can call the cell body, which is different shapes in this slide. And then they have the processes. And these processes are primarily of two types. The dendrites, which are short processes, and typically a long one, which is called the axon. And depending on the relative location of these with regard to the location of the body of the cell, then we have different types of neurons. The bipolar, of course, the two types of processes are located in different poles of the cell. The multipolar, that means that the body of the cell actually has many dendrites. It's going to be able to communicate with a number of other potential dendrites or other tissues. And then we had the pseudo-unipolar because it's kind of the body of the cell has been moved to the side. This is a typical type of neuron that you will find in the peripheral nervous system, and is a way in which these tend to be long neurons and form part of long nerves. And this is probably the most economic way of placing the body of the cell, where most of the signaling and most of the energy and most of the vesicles are going to be produced, kind of halfway between the communication between portion A and portion B for the proper transmission of the nervous signal. We could also classify the neurons, as I already mentioned, this is interesting to understand, depending on the overall direction of the impulse. As I said before, the afferent neurons are the ones which are going to be receiving the signals. They are therefore involved, they are what is called sensory neurons. These are going to be receiving the signals. They are going to receive them, and they're going to send it, as I said before, to the CNS, the central nervous system. And as I just mentioned a second ago, they are typically pseudo-unipolar neurons. The efferent, or efferent, depending also who pronounces these, are the ones that cause or are involved in the response to the signal that the afferent neurons bring there. And there are classified generally according to two subtypes. The bodies of these ones are in the CNS. And they're classified in two different types because the signal could be sent to either voluntary muscles or involuntary muscles. We have a number of muscles, an important one, which is called the heart, and that's an involuntary. We cannot do anything voluntarily to increase the speed of our heart or reduce the speed of our heart. So those are regulated by involuntary muscles, and those belong to the autonomic nervous system. And then there's the so-called interneurons, which are represented here as potential connections between the other types of neurons that I just mentioned before. And the major function is to coordinate the neural activity. So they take kind of stay in touch with the afferent and efferent, and then make a coordination of the how the transmission of the signal is taken care of. Another important difference in terms of the types of neurons is related to the so-called myelin. Myelin sheaths is something that you find in a good proportion of neurons. And this is formed by two different kinds of cells. The best known ones are the ones in the peripheral nervous system, and these are formed by something called Schwann cells. These cells are very plastic in the sense that they can kind of roll themselves around the axons of the neurons and form coverings that are equally spaced along the length of the axon. And so those are the ones that have the myelin. The myelin is represented in here like this, and each of these ones is a Schwann cell. This is in the peripheral nervous system. In the central nervous system, there are less myelinated cells, but these are done by another type of cells, glial cells called oligodendrocytes. And there are some which are non-myelinated in the sense that myelin is not formed with this kind of a structure. Many non-myelinated cells also have some Schwann cells around them, but they do not enroll themselves around to protect in this way. They simply act as a way of holding different axons together, like in a pack. Or if you look at the wires and you open one of the wires, you see different little portions inside of them. So even the non-myelinated myelinated ones have some kind of covering around them. We could also talk about different kinds of neurons with regard to the type of neurotransmitters they produce. There's a variety of them. There are many more. I want to emphasize this slide is by no means try to be comprehensive. These are some of the most important ones: acetylcholine, norepinephrine, epinephrine, serotonin, glutamate, GABA, which is easier to say GABA than to say the entire name of the particular compound, and then glycine and others. Probably the most frequently mentioned are the first ones. People call them cholinergic and adrenergic type of neurons. There's also the GABAergic, the glutamatergic. That means that they are neurons that secrete a particular compound. So let's look at what we find when we look in detail at the neuron structure. What I called before the cell body is actually more precisely called the perikaryon. This is the body of the neuron, the perikaryon. And that has to do with the space around the nucleus. You probably know the distinction between prokaryotic and eukaryotic. The karyon, again, comes from Greek, relates to the nucleus. So the perikaryon is the space around the nucleus. We're going to be talking about what's in there. I already mentioned before the dendrites. The dendrites are these processes which are short, and in the case of this neuron, which is a multipolar neuron, has many ramifications, and is allowing this neuron to interact with a number of other cells. And then we'll talk about the long process. The long process is the axon. And guess what? This one says that it has an oligodendrocyte around the axon. So this one is most likely in the central nervous system as opposed to be in the peripheral nervous system, because otherwise it would be a Schwann cell. Okay, so in the bottom part, in the peripheral nervous system, the same kind of a structure is maintained, but this time it's a Schwann cell. So remember this distinction. These Schwann cells are forming the myelin coverings in the peripheral nervous system. The oligodendrocytes, as I said before, are in the central nervous system. And then at the bottom of the picture, we find the so-called collateral branches. Some of the axons can sometimes get branched into different number of branches. And the structure at the very bottom is what is called the termini or terminals of the axons. And this is where the connection is going to be made between the axon coming from one cell and the next cell. Either it could be a neural origin or a different kind of cell. A representation of an additional axon that comes from other place is given you here and actually connects with the dendrite in this particular cell, and is forming what is called a synapse. So all these names and all these terms we're going to be repeating frequently during the next 2 hours, and we will know everything by the end before we actually even go to lunch today. It's been very formative for me to remember all these, because I knew this when I was young and innocent. Anyway, we'll discuss the innocence later. The young is obviously it's gone. All right, so let's look at what's in the perikaryon. The perikaryon is pretty much like the cytoplasm of just about any other cell. Okay, so what do you find in the cytoplasm of any other cell? You find the nucleus. Typically in the neurons, the nucleus is centrally located in the perikaryon. In fact, you see when neurons are having problems, one of the things that you see is that the nucleus is kind of pulled away, as far away as possible from the place where the problem is, and we'll deal with that later when we look at regeneration. And of course, what you find in the cytoplasm of a regular well-behaved cell is going to be the endoplasmic reticulum, it's going to be the Golgi, it's going to be the mitochondria, it's going to be a number of the typical properties in there, and we'll see later actual pictures that are going to indicate you that these things are indeed in the neurons. Now, the so-called Nissl bodies are portions of the reticular endoplasmid that contain particular types of proteins that make them stain in a different way relative to the rest of the cytoplasm. They're basophilic or basophilic, and then stain in a different way. But there is very abundant, and we'll see pictures of those in a second. Like any other cell, the neurons, and actually even more, because they are much longer than any other fibroblast or epithelial cell, they require a strong cytoskeleton organization. The main cytoskeleton components in the neurons are the so-called neurofilaments. Could be as long as 10 millimeter long, so we actually could even see them. If we were able to get them out of the neuron, we could actually see them with the naked eye. They are providing particular strong support. They are intermediate filaments. You probably remember the different classifications of filaments. There are lots of microtubules, and these are not only in the perikaryon, but are also the axon contains huge lengths of the microtubules. And in fact, it is the microtubules are working as kind of a sliding place for a number of proteins to go sliding through them, carrying whatever has to be sent from the perikaryon all the way to the synapse. All those vesicles that are carrying the neurotransmitters, we'll look into that later, are actually going to be moving using the microtubules as the sliding place to go and bring the whatever they're bringing to the end of the opposite end, the termini of the neuron. And then actin is, of course, is always a part of the cytoskeleton in just about every single cell, and is also present in there. And then there is something called, let me go ahead of myself in a way. There is the transport could go both ways. The neurotransmitters are coming to the end of the neuron, and they're going to be released at the synapse. But there is also something called retrograde transport, something that are used at the synapse and are not used anymore, and that are to be recycled, are going back for the recycling, and they go to the lysosome. Okay, and the lysosome has some residues that could be detected in there. So, as I said before, the dendrites are processes that extend from the perikaryon. They do not. They have everything else that's in the perikaryon except for the presence of Golgi. And the main function of the dendrites is to receive the stimuli. Okay. In the axon, we start looking at the axon not just here where the myelin starts. We need to look a little higher up, and because it is at this region called the axon hillock, is where most of the coordination of what's going to happen through that axon is taking place. All right, so this is a particular elongation which is not myelinated at the end of the perikaryon, that it has particular arrangement of microtubules that are going to start kind of sending everything that needs to be going down the axon. We find what is called an innexion segment, which is already part of the axon, truly part of the axon. This is not perikaryon anymore, but it is typically non-myelinated anyway. And then we find the rest of the axon that contains, of course, cytoplasm material in there. Cytoplasm material is called the axoplasm. Okay? Now, this cannot be just any cytoplasmic material. You realize that what these neurons are going to be doing is going to be channeling an electric current that is going to make things move all the way down to the axon. So in order to be able to conduce an electric current, you cannot have too many polarized, highly polarized proteins, for instance, because the current is going to stop if there is too many proteins with huge differences in charge. So typically it's more of a low protein content and contains a lot of ions that are going to be contributing to the transmission of an electric signal. All right? These collateral branches I mentioned before are these different divisions that you find in the axon itself, and then the axon terminal, which is where the connection with the next cell is going to be. They may very well characterize as those that are connecting to either other neurons or particularly with the muscle that we will look at today, and we will actually continue discussing next Monday when I come back to talk about the muscular tissue. And as I mentioned before, through this, there is a number of microtubules that are particularly involved in the transport. And the transport, again, could be what is called anterograde or retrograde. Anterograde means going towards the axon, to the end of the axon. Retrograde is coming back to the perikaryon. And there are two proteins which are specialized in carrying whatever has to be carried one way or the other. This protein, as I said, is slide, kind of slide. There is a deep molecular mechanism, hard to understand, which actually in a biochemistry class would be very useful, but in a histology class is not what we should get into. So for now, kinesin and dynein are actually, quote unquote, sliding one way or the other with the help and along the microtubules inside the axons. So now we're going to be looking a little more in detail at the perikaryon itself. And we find things that we have mentioned. And one of them is the so-called axon hillock, which is, there is a tiny circle indicating where it is located. It's this prolongation of the perikaryon where the axon starts. And it's characterized primarily by, it's a place where the macrotubules are organizing themselves, forming bundles, and those are going to be going down into the axon portion. Typically, the other components in here, particularly the nissl bodies, are not present in that particular region. The next one, as I mentioned a second ago, is the initial segment, which would be immediately below the axon hillock. Is not subject to myelination, and is where however, the stimulus is integrated. It's like, let's say, where the computer is placed inside the neuron. Once the neuron is stimulated, the signal has to be integrated somewhere and with the physiology and the mechanisms of the cell. And that is in that particular segment, which is not insulated. Myelin, one of the things that myelin does is produces a huge insulation around the axon. This is not insulated, probably because of the reception of the stimulus requires more accessibility at that particular point. The collateral branches and transport we have already talked about, but I wanted to show a picture of what the perikaryon looks like with the nucleus in the middle. And by the way, one of the things that you also find very frequently in this is, in addition to the endoplasmic reticulum, is a large amount of mitochondria. You can find mitochondria also along the axon sometimes, and particularly close to the synapses, because these processes consume energy. So let's talk about the axon. The axonal transport, as I said, it could go both ways. It could go towards the axon, towards the terminal or the synapse, wherever it's going, and it could come back with a number of different kind of materials. Primarily, the anterograde transport is going to be bringing whatever has to be secreted at the synapse. So primarily, the main transport is vesicles that contain the proper type of neurotransmitter, and is going, as I said, quote-unquote, sliding on the microtubule bundles, which are located inside the axon. After what the function of the synapse has been accomplished, some of these vesicles are actually going to be recycled back, and other portions and other components of the cytoskeleton, and these are going to be taken by in the opposite direction by a mechanism which is very similar, although the two proteins are very different structurally speaking. The mechanism is very similar in the retrograde orientation. Now, the proper, as you probably realize, the proper functioning of the transport in one way and the other is essential for the life of the neuron. And in fact, there is a very well-known disease because of the guy who had it, and it's called Lou Gehrig's disease. Who knows about Lou Gehrig? Almost everyone here in the room knows about Lou Gehrig. There you go. You're Yankees. So this is called ALS, amyotrophic lateral sclerosis, and this is caused by a defect in axonal transport. All right? So it's not just that the things go back and forth and this is like we're in a fair, and no. If this doesn't work and you're not properly regulated, things don't work the way they should, and the nervous impulse doesn't go, and then the muscles don't start not working, and you die. That's a very short description of the disease. But so in terms of the anterograde transport, you need to actually realize that there's different levels of the regulation requires for two different kinds of levels of speed. There is a kind of slow mechanism, which is either 1 to 4 millimeters per day, and this happens for the growth of the axon when the axons are being in development. And then there is also the fast stream. That's the one that I've been talking about primarily about the moving the vesicles carrying the neurotransmitters, and this is substantially faster than the other one. We go from 1 to 4 to more than 10-fold faster compared to the slow version of this. And this, as I said, is the one that is responsible for the transport of the neurotransmitters, which are essential for the functioning of the nervous system. And again, the protein that's involved in the anterograde is kinesin, and the protein that is involved in the retrograde is called dynein. And the retrograde typically goes kind of in between in terms of speed. It is intermediate stream and is between 10 and 100 millimeters per day, and is considered a salvage pathway because it's simply primarily devoted to recycling materials, recycling things that have been used, have been used for a purpose, and now they're going back and are going to be recycled and probably used again for something else. And so it transports recycled portions of the membrane that was involved in the formation of the secretory vesicles and also cytoskeletal components. So we get to try to understand what the synapse is. I've been mentioning the word synapse a few times, and the synapse is basically the point or the structure that establishes a connection. Let's talk about just the nervous system today. We'll look at the synapse with the neurons with the muscle later and next Monday, but pretty much is a structure that allows the transmission of the signal from one neuron to the next nervous cell in the system. For the time being, forget about the names of the enzymes involved in there because this is, again, not biochemistry. This is about the nervous system. And so what we have is two membranes. There's a structure that contains two different membranes, one coming from the axon that carries the signal, and another one that it has the proper receptors which are going to be receiving the signal. There is, of course, specificity, because if the neurotransmitter is of the one particular type, it has to be a specific receptor receiving that kind of a neurotransmitter. If the receptor is a different one, there's going to be response on this side. So we'll see later how this whole process is, but I'm going to try to identify that there's primarily two kinds of synapses. The synapses are the so-called chemicals, which are basically based on the secreted neurotransmitter binding these receptors and stimulating a couple of signaling pathways. And there is certain space between these two membranes. The electrical type of synapse, the space is much more reduced. I don't know what you guys, I imagine you guys have studied the epithelial tissues already, and you know about the so-called gap junctions. Gap proteins, there's a family of proteins called connexins, and those are the ones that maintain much more tight interaction between the two molecules, the two cells in there. And in fact, it is through the little pores of the connexins that some of the signal is transmitted. All right. So there is no need for this space in the chemical synapses is larger, is greater, allow for the diffusion of the neurotransmitter into this space, finally reaches the receptor in that way. And there is some synapses that are actually of what is called mixed type, because combining both, you can find that certain synapses allow for the transmission of neurotransmitters and for the stimulus, electrical stimulus done through gap junction mechanisms. So talking about the synapses and how they work, this is again two different versions of the picture compared to the other ones. The vesicles are going to be carrying a variety of things. This need to send the neurotransmitters. This is of course talking about the chemical synapses. They could also be releasing certain hormones, and one of the main important elements in the synapses is that the depolarization, and we'll get into that very soon, has to be transmitted. In other words, the electrical alteration caused in these cells by the whatever was the signal, it has to be transmitted, and calcium plays an important role in terms of the overall process and the steps of the transmission of the signal. So what happens is that the presence of calcium is going to be actually what triggers the exocytosis, and the transmission of everything down to the end of the axon is done by depolarization. And we'll talk about those two things in just a second. In terms of the synapses, again, I mentioned that morphologists and histologists tend to classify everything as many types as they can, to assign names to things. And of course, you have the possibility that an axon binds either a dendrite or the cell body of, let's say, a muscle tissue. And also it could be that an axon binds another axon, which is in the same terminal region in there, and there is a transmission like the one depicted in here. So depending on where the final interaction happens, people use these names. The axodendritic, which means the axon goes to the dendrite, to the next cell. The axosomatic, which means that it goes to a cell that is not of the neural type. The dendrodendritic, which means dendrites could actually interact with the dendrites themselves. And then the axoaxonic, which is this particular one in here. So this is something for you guys to understand and to know. But I don't think the world will disappear if you forgot what these names indicate. Okay, so in terms of the structure of the synapse, we talk about three things. I told you before that there is two membranes, very specialized membranes in terms of what they are and how they work. And this membrane here, the one in the axon that's coming in, and we'll look at what the meaning of the 1, 2, 3, 4, 5, 6 is later. This is specialized is called the presynaptic membrane, and it's primarily the place that contains all the signaling necessary to favor the exocytosis. The membranes that are bringing the neurotransmitter are going to be fusing to the cell membrane, and they're going to open up and releasing that particular neurotransmitter. Then the space in between that I said in a chemical synapses, it tends to be larger than in the electrical synapse, is what is called the synaptic cleft. And the postsynaptic membrane is the membrane that is receiving the stimulus, and it typically contains a number of receptors that are specialized in receiving either the neurotransmitter or changes in the concentration of sodium, and we'll see in a few minutes what the changes in the concentration of sodium are doing in terms of causing the depolarization and the transmission of the signal itself. So presynaptic membrane, postsynaptic membrane, and synaptic cleft. These are words that you will find, and we'll be talking about at different points. So if you want to look at, I just wanted to show you some examples of the way they look in a histological preparation. It's very easy to distinguish where the presynaptic membranes are. Anyone can guess why it's easy to detect where the presynaptic portions are? Where are the vesicles? You recognize them by the vesicles. The recipient, this is the synapse. Okay, this space in between is the synaptic cleft. They specialized structures on both ends. Here is the same situation. Okay, so you can see that the pre-synaptic membrane is loaded with different kinds of vesicles, and there's going to be activation of this cell for whatever particular purpose this cell system is involved in. This is an example of what we will look at more in detail next Monday when we look at the interaction between the nervous system and the muscles. The peripheral system, one of the easiest ways to understand how it works is the reflexes. Okay, imagine that these were hot and I happen to put my hand on top, the immediate reaction is a reflex arc. It's sensitive in my hand because of the sensitivity to heat is going to force me to move my muscles. The sensitivity comes from the nervous system. The ability to move my arm comes from the muscles, and there is an important interaction between the two. This is an axon that has been cut transversally there. You can see the Schwann cell that still forms the myelin of this. So because of this, we know, and because of the connection with the muscle, we know that this is the peripheral nervous system. Because this is one cell. If it was a central nervous system, it would be an oligodendrocyte. And then you can see what's inside the axon. There's mitochondria, there is plenty of vesicles, and then in the particular case of the muscle, they form a very architectonically interesting, if you want, anatomically interesting structure, which is called the motor end plate, that facilitates the transmission of the signal to the muscle. This is the other kind of muscle. This is smooth muscle. This is a type of muscle that is involved in non-voluntary contractions and is present in many tissues and in many places. And the interaction is slightly less impressive, if you want, from a histological point of view, but the thing in common is the presence of vesicles, which are, this is a nerve ending, and again, you can tell where the presynaptic portion is by the amount of the vesicles that appear in there. All right, so how is this famous electric impulse being generated, and how is it transmitted from wherever it starts to the end of the axon? We need to discuss a few things. We need to talk about proteins which are part of the membranes and that have specialized functions. So some of them are called pumps, some of them are called ion channels. And we need to understand the difference between the so-called resting potential, the action potential, the saltatory conduction, and we need to understand how is the actual overall transmission being accomplished. So let's look at the membrane. Of course, the membrane, everybody knows, is formed with it's a lipid bilayer, and some of the proteins which are in there, intrinsic proteins, have the function of transporting different kinds of things from inside to the outside of the cell. This is particularly a sodium-potassium pump. Is actually a mechanism consumes energy. Therefore, that's why the ATP is actually converted into a hydrolyzed in the process. And this kind of pump, for every 3 sodiums that go out, brings 2 inside. So if under normal physiological conditions, what happens is that for every 2 potassiums inside, there is 3 sodiums outside. That creates a difference in charge. There is more positive outside and there is more negative inside. So an electric field is always there, and the membrane is what is called polarized. Okay, so there's a dipole. It's a positive pole and a negative pole. And it's simply maintained by the amount of sodium outside being much larger than the amount of potassium inside. Last time I discussed this with somebody in a different context, people were wondering how could it be negative inside if there is potassium, which is a positively charged ion anyway. Inside the cell, there's many other things in addition to just potassium, and the potassium is quote unquote, neutralized, if you want, partly by binding to a number of negatively charged proteins. Okay, so that the fact is that there is a differential in the two poles, and the, outside the charge is mainly positive. The net charge inside is negative on both sides of the membrane. So there is a number of ion channels also, in addition to the pumps, that are involved in maintaining this status. Some of them are specialized on potassium, others are specialized in sodium. The sodium ion channels could be just turned on, let's say, by changes in voltage. The other can be turned off by binding of a ligand to the outside extracellular portion of the molecules that form the channel, and others can be activated by is what is called signal gated. It comes from inside, ligand gated if it comes from outside, or voltage gated if it is a difference in a change in the polarization of the membrane. There is always this potassium specialized channels that are actually allowing certain small amounts of potassium always diffusing to the outside. There is no expense in energy in any of these cases, but there is a way of maintaining the equilibrium. And there is more channels specialized in sodium than in potassium, letting you know that it's actually important to maintain these, that the sodium is pumped out as opposed to the sodium coming in. So the difference in terms of the concentrations that I'm talking about are this is the internal, this is the internal portion of the cell, this is the inside of the cell, this is the outside of the cell. For potassium, the outside is in the order of 4 millimolar, the sodium is 150 millimolar. Inside, the potassium is 140 millimolar, and typically 12 millimolar only for the sodium. The chlorine ions that actually probably could form part of both molecules do not contribute too much to the differential in the polarization of the membrane. So, but this is important to remember: sodium outside, potassium inside, polarization of the membrane. In fact, if you are to measure the voltage difference between the inside and the outside, you can see that is a difference of about. You can measure that by using microelectrodes. In the membranes, you can actually put one of the electrodes inside the axon and the other in the medium outside, and you can see that under resting conditions, the neuron is not stimulated. The voltage difference that is found is what is called equivalent to minus 70 millivolts. All right? And this is what is called the typical voltage at the so-called resting state, and this is what we call the resting potential. All right? So under no stimulation, the resting potential for a well-behaved neuron is in the order of minus 60, minus 70 millivolts. Now, if there is a stimulation of some sort, the potential is the equilibrium between the sodiums and potassiums is changed. The sodium, the channels open, or the pump works in the direction of getting breaking the equilibrium also, and we go into a phase which is called depolarization. The original standard polarization is modified as a consequence of the entry of sodium inside the cell. So we reach a point of maximum what is called action potential. All right? This action potential could be in the order of plus 30 millivolts. So we have gone from minus 70 to plus 30 is a whole 100 millivolt difference that we undergo just simply because of the stimulation of the neuron. All right? After that stimulation, and we'll see how this is going to happen along the axons, everything goes back to normal. One of the beauty things about most processes under physiological conditions in biology is that everything that's important tends to be reversible, tends to go back to normal. If you look at, I don't know how much you guys know about these kinds of things, but now it's very fashionable to talk about epigenetics. Okay, you guys also know that science goes by fashion. Now it's epigenetics. There will be the CRISPR-Cas system. There will be some other things like that. But the epigenetic signal, if these signals were not transient, we would be in deep trouble. If the depolarization polarization of the neurons were not reversible, then we will be excited all the time. The neurons will be excited all the time, and that will be incompatible with the normal physiology. So cells tend to recover the original state, and in doing so, they go through a process called repolarization. And the repolarization is kind of they overdo it. Cells tend to overdo it a little bit. Let's put it this way: and they go into a phase which is actually brings the voltage below the normal resting potential of the cell. And this is what is called hyperpolarization. Is classified here or qualified here as an undershoot. Is inertia. Okay, if we all start running, we tend to not stop immediately. We run a little bit longer. That's what happens, and then it recovers, and slowly it's going to go to the resting membrane potential that is reached again. And here is the way in which this cell was excited. We went from the resting potential, action potential, a hyperpolarization, and we recover everything. And what's important to realize also is the scale. We are talking about milliseconds, okay? So a neuron can be stimulated and goes back to normal within 7 seconds or less, because if we consider this, the point in which it goes back to normal, we're talking about 4 milliseconds. So this is tremendous speed of the stimulus and how it has to be transmitted. So what happens is that there is a coordination between, this is another way of representing the same thing. If this is the depolarization of the cell after a certain stimulation, this is the resting potential, then the cell gets stimulated. And what this happens is that it is coordinated with what happened at the level of the sodium channels. The sodium channels are initially closed under resting conditions. There is it's a depolarization, the channels open. Once the channels are open and a stimulus is created, these channels are so-called inactivated. In another word, they are not working as efficiently as they should at this point, and then finally they go to being closed. So within the same length of time, this is now 2 milliseconds that we're talking about, the channels go from closed to open to now we need to start kind of coming down to being closed again. Yes? What brings things back to normal is more the potassium movements than the sodium movement. So it's possible that it's involved in the hyperpolarization. All right, so this is another picture of this, and what you can see is that under normal conditions, when the charges are positive in the outside and negative in the inside, the membrane is therefore polarized. The the ion channels are closed. Then there is the depolarization. They open initially. They finally assume a structure that is incompatible with the transmission of the channeling of the ions and goes back to normal. So there has to be, again, a perfect coordination between the length of the signal, this intensity of the signal, and the duration of the opening and closing phases of the particular ion channels. So what happens and how is it transmitted? Well, the way it is transmitted, theoretically, it could actually go this way or that way. The depolarization, if it happens here, it could theoretically go in that way or go in that way. But what is being demonstrated is that in axons, it always goes from the pericardium all the way to the axon terminals. All right? And the way it's done is that there is a depolarization early in the axon that changes now the inside of the membrane becomes positive, and then this sodium that has come in, it starts diffusing with sufficient speed to maintain that kind of a wave of depolarization. As the sodium is increasing in here, there is a depolarization immediately following, which is depicted over this slide tries to depict, and the movement is in this particular direction, and things are going back to normal as the a wave of depolarization moves on. A segment will be repolarizing, and then another segment has gone back all the way to resting state. Okay, so it's a wave of depolarization that happens all along the axon that carries the nerve impulse from where it was initiated in response to whatever orders the cell gave in the physiological process in which it is involved, all the way to the axon to bring the polarization to that location and allow the performance of the function that the axon has to do. Now, one of the things that we need to understand is that there is a way of actually even accelerating the way in which this is transmitted. And in that regard, myelin helps greatly. Okay? Remember that I described before that myelin is simply a structure that is formed by having, typically in the peripheral nervous system, a Schwann cell kind of wrapping a number of times around the axon. And it goes from this kind of initial structure to form something tightly dense packed, sheets of the Schwann cell that forms the myelin. Okay? The Schwann cell retains the integrity. It forms all these wrappings around it, but you can still see that it has a nucleus and it has a cytoplasm. But what it does is actually insulates the axon from any electrical changes that happen outside the nervous cell, the neuron. But it also, they wrap around the axon at certain intervals, okay? And there's spaces where there is no Schwann cells located, all right? So what this allows the cell is to get into a kind of conductivity. This is the portion in which there is no Schwann cell wrapping around the axon. This is the axon here. And these places are particularly specialized places called the nodes of Ranvier. Okay? And these nodes of Ranvier are in this type of neurons, which are myelinated, is the only places where depolarization happens. So depolarization happens here, and the distance between two nodes of Ranvier is such that the entry of the sodium at this point is going to allow it to diffuse quickly enough to this other place and cause the wave of polarization to be transmitted. And because of this is how we call this saltatory conduction as opposed to diffuse conduction. What this means is that you have two axons, same length. One has the myelin and the other one doesn't have it. The impulse can be moving much faster in the myelinated ones than in the non-myelinated ones, all right? So if you have myelinated neurons are going to be much more specialized and conducting quickly relative to the non-myelinated ones. And this is what I try to show you guys with this slide. This is passive conduction is this typical wave of transmission that I mentioned before, and the saltatory conduction is actually going to go from one place to the next, and it's going to take much shorter. So the signal appears to jump from one place to another, but conduction by this mechanism is about 30 times faster than what happens in a non-myelinated axon. So the depolarization has gone all the way through the axon one way or another, and it reaches the synapse. And what we find is that once the action potential has reached this location, then there is an entry. I promised you that I was going to tell you what the 1, 2, 3, 4, 5 secret numbers were in the previous slide, and here is where they are. So once the depolarization, the action potential comes to this stage, there is entry of calcium. This entry of calcium promotes the movement of these vesicles to the docking places, docking proteins that specialize in the presynaptic membrane, and the release of the neurotransmitter, and then the activation of the signal is transferred to the postsynaptic membrane and through the receptors or through the gap junctions. If it were an electric synapse, instead of having all the secretion, the voltage, the action potential will actually reach the connections, and that will allow the exchange of small molecular weight compounds. And the final of the story is that there's been a transmission of the signal from one cell to the next. Yes, sir. On the previous slide. The action potential is the electric signal. It's a change in the electrical situation of the membranes. The speed, okay, well, there is a coordination. Nothing happens, okay, but it's not responsible immediately directly. You can actually, you need certain level of modification of the action potential to start pushing things through, all right? So the vesicles pretty much travel in a coordinated way, but it's not really responsible for the movement itself. It allows the situation in the cell, in that particular portion of the cell, to change, and the cell, what we needed to do is actually look at the molecular mechanism of transport in detail, but I wanted to stay away from that. Okay. All right. So then at the this is a couple of pictures of two different kinds of junctions. Remember that I talked about different neurons secreting different neurotransmitters. This is the cholinergic ones that secrete acetylcholine. These are the ones that secrete noradrenaline. And this is two different types, but the structure is pretty much the same. And there is always the secretion that is promoted by the activity of calcium. Calcium is the one that is important in terms of triggering the whole mechanism of secretion. So don't go too far away. There's more that we need to talk about the nervous tissue, but we can probably take like a 5-minute break, bathroom break, drinking break, whatever break, and we'll get together again in 5 minutes. Thank you. I can put it on.
Yeah.
You want me to use this? I don't have it. We can put it in. Just, sure. So this goes in a pocket or someplace, or right here?
Yeah. Just turn on the switch.
And then we can put it.
Yeah, right. That should be.
Is this working?
Yeah, scratch it. I think it should. Yeah. If you just lift it higher, closer, I think it'll pick up.
All right, guys. Let's go back to the nervous system again. Now, can you hear me better? All right. Now I'm mic'd and everything. Now I feel like one of those important people. Let's go to the second part. And in this second part, of course, we also have a number of learning objectives. And we want to look at the nervous system as a system. And then we're going to go down to studying some of the types. We have concentrated a lot on the neurons. We're going to be looking at some of the types of cells, the glial or glial cells that I mentioned before. And I want to put them from the perspective of protective systems of the nervous system. So we were looking at a number of protective devices that are there, including the, why this is not a larger arrow, I don't know, but this is called a mutation. If you have ever seen a mutation, now you can see it. There you go. There you go. Magic. It must be the microphone. So we have the glial cells or glial cells, which are going to be primarily protective. We had to talk about the meninges, which are membranes that are going to provide a protective function for our brains. We're going to be talking about the blood-brain barrier, which is going to prevent things that should not get into our brain from getting into our brain. And then we're going to talk about the CSP F, the cerebrospinal fluid, which is going to provide kind of a mechanism to protect us also from potential injuries. And then we'll concentrate on the peripheral nervous system, try to understand how it works. And in particular, we spend some time looking at the erroneously named autonomous nervous system, because it's really non-autonomous. It depends on the brain or the spinal cord. But it's a portion of the type of a peripheral nervous system that deals with the non-voluntary acts that we cannot regulate. So everything is being organized to answer those questions. So if we look at the central nervous system, we have the cerebrum and the cerebellum, which are part of the inside the skull, and then the spinal cord, which is inside our spine. And those are the main structures that play fundamental roles in how we interact with the environment and how the things are coordinated. So if we look at the brain itself, we have three main portions in there: the cerebrum, okay, the cerebellum, and the brain stem. The cerebellum is something that every time I help my wife prepare cauliflower for dinner, it reminds me very much of my cerebellum. So I treat it with a lot of care. So each of these portions of the brain have different functions, and they're actually within each of the portions. There are very specialized subsection or subdivisions that specialize on different things. It's the largest portion of our brain, the cerebrum, and it has the ability in the regions and the neurons and the connections to allow us pretty much to think, to rationalize, to get in trouble, and things like those. The cerebral cortex is where we do most of our computing, let's put it this way, and then there is, as we go inside, we find a number of the so-called basal ganglia. We'll look into what the ganglia are a little later, and these are related to the synchronization of movements. The hippocampus, it has to do with long-term memory, and the so-called amygdala has to do with memory and emotional behavior. Of course, we are not going to get into more than that, deeper than that, except we're going to be looking at what kind of cells we find in different places. The cerebellum is located, as I said, kind of behind in the occipital region of the brain, and its consistency is the size of a golf ball, and the consistency and the looks of a cauliflower, and is actually very important in coordination of movement. So fundamental for that point of view. And then in the brain stem, we find also different divisions. Remember that histologists and anatomists like to give names to different portions. One of them is called midbrain. That is kind of the portion here where it really interacts, is actually in orange, would really interact with the cerebrum. And it has to do with the reflexes and based on hearing and sight. The brain stem, this one here, this portion, is the beginning of the spinal cord, and there is a bulge that you can observe in it, and this is the so-called pons, and the deeper portion of the brain stem is called the medulla, and it's involved in different physiological functions related to breathing, heart rate, digestion, and so on and so forth. So if we look at the spinal cord, which is the next thing down in the central nervous system, we have to start distinguishing, and in fact we could distinguish that in here, in this portion of which is cut of the cerebrum. You can see this, it's not just by accident that this has two different colors, okay? And in the spinal cord, there is also two different colors in here, and we have to start distinguishing what is called gray matter and white matter. And basically, we'll see pictures later that will make you wonder why we call them this way, because if you stain them with different stains, you can actually even reverse the way they look. But forever, the names that have been assigned to these regions are based on the initial stains that were developed, and that's why they talk about the gray matter and the white matter. What do we find in the spinal cord is there is a portion very well defined inside that has the shape of an H. That has the so-called horns, the legs and the arms of the age. They're called the posterior horns and the anterior horns, and there is a portion in there that if it gets together, these two horns in the middle, is called the central canal. All right. What you find in this region within the age is what is called the gray matter, and contains primarily the neurons, the pericardions, the nerve, the cell bodies are around here. You can see them, and there is plenty of nerve fibers. Nerve fibers are made by bundles of axons themselves, and there is also glial cells that are going to be providing support for those nerve fibers. If you go to the so-called white matter, what you find is primarily glial cells, and there are no neuronal cell bodies. And then this usually contains plenty of myelinated axons packed together in the so-called tracks. All right? But there are very well differentiated divisions within the spinal cord. And as I said, realize now, we'll see what the difference is with other regions of the central nervous system, that in the spinal cord, the white matter is outside, and I was going to say the dark matter. The gray matter is, the dark force, that, from that kind of generation. The gray matter is inside. We looked at other portions of the brain later, other portions of the CNS, where it's actually going to be the reverse. That we'll see the difference. And that simply has to do, the composition primarily remains the same in terms of what you find in the gray or in the white matters, but is the distribution and the functionality of that particular segment that is different. And of course, one of the first cases that we find that is in the cerebrum. Now, this is a section of the cerebrum. Is indicated in here. The cerebral cortex is the most outside layer of the cerebrum, and you can find now that the gray matter is outside and the white matter is inside. But what you find is essentially the same thing. You find neurons and glial cells in the gray matter, exactly as we saw in the previous case, and in the white matter, what you find is nerve fibers. Okay, this is axons packed together, maintained by glial cells, and in the cortex is the place where the higher thought processes and the coordination of movements takes place. But I just wanted to show you the distribution of those two. When you look under the microscope, and this I should make a parenthesis, I'm originally from Spain, and there's this guy named Santiago Ramón y Cajal, who got the Nobel Prize with another guy from Italy called Renato Golgi, and we didn't care too much about him in Spain. But they did that because primarily Ramón y Cajal did is the main contribution to establish the nature of these structures of the brain. And if you had the time and you ever want to go to Spain, visit his museum, and you are going to be amazed by the kind of microscope that this guy was using, and he was able to see these things. I mean, and also he was a great artist, he was able to draw everything. And now with the super new microscopes and everything we have, everything has been demonstrated to be the case. So the guy was not lying. So, and as I was mentioned before, in the cortex, you can see that there is a reversion. That's my main point here. There is a reversion of the gray and white areas. And depending on, of course, it's composed by cell bodies and nerve fibers. And you can distinguish of low magnification 6 different layers. They receive different names: the molecular, outer granular layer, the pyramidal cells, the inner granular, the inner pyramidal cells, and the polymorphic cells. But if you do high magnification, then basically you can see actually 2 layers, which are called the superficial, which would be the main top, and the pyramidal cells, which is what most of these large bodies of cells are located in here. And the axons, many axons, extend all the way from the top to the bottom of the cortex. So the superficial layer, as you can see here, in this portion, most everything you see are axons, all right? And then in the pyramidal layer, what you see is that it's primarily cell bodies are appearing in here, and you can see some of the axons actually go through all the way to the bottom and to inner portions of the cerebrum. In the cerebellum, the situation is the same. As you can immediately see, the The gray matter is in the outside and the white matter is in the inside, and the composition remains the same. The gray matter contains neurons, primarily neural bodies and glial cells. The white matter primarily nerve fibers and glial cells, and the function of these, as I said before, is the coordination of movements. So if we look in more detail, and again, this looks very much like some of those drawings from Santiago Ramón y Cajal, with the silver staining. By the way, he was the first one to apply silver staining to the analysis, histological studies of the brain. You can distinguish fundamentally three kind of layers. This is, of course, the surface, and we go towards the inside. We find the molecular layer, which is similar to the one that we saw in the cortex, and what you find is pretty much a region that, depending on the staining, that's something that I tried to mention also, that what you see or what you don't see in histology is extremely dependent on the stain that you use. And we'll see in the lab, I'll show you two different pictures of the spinal cord, and you'll see the huge difference between the two stains. But what you can see here clearly is that this is a region of what they call molecular layer, primarily contains axons. Then in the granular layer, we find is a very peculiar type of cell called the Purkinje's cell. In those days, scientists used to assign their own name to different things. So Purkinje is a guy who identified these neurons, and the same way that Golgi gave the name to the apparatus inside the cell, the Golgi apparatus. You want people to remember. It's the tendency of humans to try to become immortal. And then, this is histology, not philosophy class. I will talk about the philosophy later after the class, and maybe in the bar. Below the Purkinje cell layer, you can find a really high density layer in terms of the numbers of cells, and this is the so-called granular layer. So there's huge amounts of cells which are located in there. And there are, the difference is that in the molecular layer, and this is important to realize, the nerve fibers are unmyelinated. That in the Purkinje cells, of course, are different from the rest of the cells. And then in the granular layer is multipolar, small multipolar neurons that are particularly located in there. So let's look now start looking at the different protective systems that exist in the nervous system to protect the key component, which are the neurons. And one of the types of protective mechanisms is the use of the glial cells to kind of hold them together and insulate them from other electrical fields and things like those. So in the glial cells, there are different types. The oligodendrocytes are also very similar to this, and I already said that several times, to the Schwann cells. They're very plastic, and they can kind of roll around the axons and form the equivalent to the myelin in the central nervous system. They can actually wrap, and this here is an example in this portion of the picture. One oligodendrocyte can actually wrap more than one axon and maintain the structure together. So these two nerves these two axons are within the same proximal space. There is the astrocytes, and they call astrocytes because they look pretty much like a starfish, with different versions. And there are two different types. The fibrous astrocytes, which are always only in the white matter, and the protoplasmic astrocytes, which are in the gray matter. So it's a specialization in terms not only you find differences not only the way they look, and we'll see pictures of those, but also in the way and where they are located, and therefore with other cells or axons they interact. Actually, by the way, I just realized that this is an astrocyte, and this is the one I mentioned. I tried to point with the oligodendrocytes. And then we have these kinds of cells, which are the microglia, and they are like tiny cell bodies with huge amount of processes. They are very small. That's what I call the microglia. They're much smaller than the other cells. And the microglia are could be stimulated to become like the immune systems, like the they can actually even work like the even phagocyte some molecules and behave pretty much like components of the immune system of the brain. And then we have these other cells that look pretty much like epithelial looking. Is a single layer of epithelial looking cells, and these are the ependymal cells, which are lining certain components of the brain and the spinal cord, and they are involved in the secretion of the cerebrospinal fluid that I mentioned before. All right? So these are the four main types, and then we'll talk about properties and characteristics of each of them. As I repeated myself several times, and probably because I consider this important for some unknown reason, the oligodendrocytes are able to roll themselves around and bind tightly to different kinds of cells, and in fact form structures very similar to the myelin sheaths, which are typically formed in the central nervous system, as opposed to the Schwann cells, which are in the peripheral system. They all spaces. They also leave spaces, like creating the node of Ranviers. So the overall structure remains the same. The difference is that one type of cell works in the peripheral nervous system, the other one works in the central nervous system. This is a cell that can myelinate many axons, as I said before. Myelination is actually something that takes place during the development of the individuals, and this is interesting to know also, because the insulation of the nerves is not completed by the time individuals are born. It's actually something that develops over time. And these cells, because they are so huge and so complex, they do not proliferate, for instance, with the speed. They do not divide with the speed, let's say, of a fibroblast, or least of all of some of the cells in the blood. Some of the blood cells that proliferate very efficiently, or even the skin cells, which are relatively slow, but still maintain level of proliferation continuously. The astrocytes are, as I said earlier, something that looks pretty much like a starfish. All right, is a centrally located body with many processes that go outside, and these astrocytes could actually bind either axons or even could bind and protect some of the vessels, blood vessels, which are located inside the central nervous system. They could perform different functions, and as I said, the main one is protecting the cells, and they can contribute to the removal of some of the residues of the transmission of the signal, and in particular, they have been called, one of the functions that have been mentioned about these cells is that they can actually soak some of the potassium that is left over after the signal, the creation of the potential and the transmission down the axons. As I said before, the astrocytes can be protoplasmic in fibers, and important to realize is that the function might be associated with particular interaction with particular cell types, because the protoplasmic are only found in gray matter, and the fibers are only found in white matter. And the difference between one type and the other is simply the amount of processes that one has that is much more abundant in the fibers than in the protoplasmic. The microglia, as I mentioned before, could be very small cells, much smaller. They can be found everywhere, as opposed to the other types of astrocytes. You can find them in the gray matter and the white matter. They are most of the time inactive unless they're called on duty, and when they're called on duty, become active in terms of protecting by removing and phagocytizing, involving phagocytosis of something that is not supposed to be there, what it is. So it's pretty much a role equivalent in the brain or in the central nervous system to the macrophages or macrophages to in the immune system. Okay, they can behave when activated. They become what is called reactive microglia and defend everyone. And then, as I mentioned, the ependymal cells are simply a single layer of epithelial-looking cells that contain actually many of the protein and structures that are typical for those, the gap junction, the tight junctions, the desmosomes, and all that, form a tight layer because one of the things that they do is they line the places where the cerebrospinal fluid is going to be flowing around. So you don't want spaces in between where the cerebrospinal fluid can actually leak out of the place. So in a way, they also resemble the epithelial cells lining certain other organs because they have the so-called cilia or cilia. Actually, there should have been an E in here to make it perfect Latin. And therefore, what they do is they keep moving the cilia in the surface to maintain the movement of the circulation of the cerebrospinal fluid wherever it's supposed to be going. So in a way, it could be a single layer of epithelial cells that actually secrete the fluid and could maintain it from running into other tissues and becomes an impermeable layer used in this way. A different level of protection is done by the so-called meninges, and these are immediately under the skull. There are 3 layers we looked at what they contain and what their structure is, that are kind of covering the brain and the central nervous system. And what these guys are doing is simply preventing any damage of facilitating the distribution of blood to the top layers of the brain and creating a space and something that we'll look into this segment here, creating something that is going to, let's say, in case you have you bump your head against something, it's going to prevent. It's going to have like a little rubbery system that is going to prevent you from hitting directly into the brain itself. And there is they contribute a system of protection. There are 3 layers, so-called dura mater, arachnoid, and pia mater, and we're going to look into what they do. The dura mater is the most external one. It's the one that's immediately under the skull, under the bone. It's actually in touch with the bone. It's connective tissue. This is, remember that we needed to talk about the connective coverings in one of the very early slides. Is close to the cranial bones, and is very well vascularized. If any one of you guys had any cut in the brain or something that had broken or having some accident, you see that there is huge amount of blood coming just from this epidermis itself, from the skin itself, but also there is a huge amount of blood that could come out that way. So that's the one that's closer to the bone. The next is the arachnoid, which is a very interesting structure. It again is connective tissue, but is specialized in forming these type of columns, all right, which are the so-called trabeculae. This is in Latin now here. This is a plural of trabecula, all right? And what this thing does is imagine a spring, okay? So if you bump your head, this acts as a spring that can be flattened and therefore prevents the actual brain to hit the hard stuff that is forming the skull. And this space that is in between here, just in case if these were not enough, is actually full of the liquid. Okay, this is space that is created here because of the particular structure of the arachnoid membranes is actually full of cerebrospinal fluid that provides another layer of protection, and is also highly vascularized, and you can find plenty of vessels which are lined by this kind of tissue. And then finally, towards the inside is the most inner portion of the meninges is the so-called pia mater, and this again is mainly collagen fibers. Okay, so this is it's not as highly cellular as the other ones are, and it contains lots of collagen that you have been hearing about collagen for. At least they were talking about collagen when I came last time. They were talking about the bone and a bunch of other things. And is also lining all the pia mater is this thin layer here. See, and that is actually lining the spaces where the blood vessels are going into towards the brain. The blood-brain barrier is actually combination of glial cells and endothelial cells, because this is something that happens is to prevent certain types of substances to go into the brain. And this is mainly a chemical barrier, if you want. The role is fundamental, but unfortunately is a problem if you want to deliver things to the brain. If you have a brain tumor and you want to deliver certain chemotherapeutic agents, they do not go to the brain barrier. And if you manage to permeabilize the blood-brain barrier so that make things easy, go in and out more easily, you run the risk of having things that should not be going there and making the problem worse. There is a number of studies now to see how one could do this transiently, very quickly, in such a way that there is no problem. You get in what you want to get in, but nothing kind of comes out, leaks out, and creates a larger problem. But the research is still not as the level of where we could use this routinely. It's easier to depolarize any membrane, and in fact we do this in the laboratory all the time when we want to get DNA, let's say, into cells for whatever our purpose is in research. We use one of the procedures is called electroporation. I call it the electric chair for cell work, because basically what you do is you put your cells in a cuvette that has the two electrodes, turn on the machine, and what it does is actually changes the polarity in one split second, and changes the polarization of the polarity of the membrane, and the DNA, which is negatively charged, now it has an easier chance to go inside as opposed to being outside stuck to the positive charges in the outside of the membrane. All right. So if that could be done at the level of changing the permeability of the blood-brain barrier just for a split second, so your drug goes in and cures the tumor, that could be great. But as I said, we are not still in a position to do that. The cerebrospinal fluid is again providing some an extra layer of protection, because it's like certain regions in the nervous system are kind of floating and being surrounded by water. So you can move when you are in the water. You can move and maybe displace the water more in one direction than in the other. But that also helps preventing a shock that could be caused by some sort of trauma. So it has to is a liquid, which is simple, is not rich in proteins. You don't want a liquid which is way too dense. That could actually be creating problems by clogging some of the circulation systems. Contains a number of ions, and it keeps circulating, and is produced primarily in a region that is inside the cerebral ventricles. I'll show you pictures of the cerebral ventricles, which are primarily lined with layers of these ependymal cells and some epithelial cells mixed with them that form these structures. Okay, this is the choroid plexus. You see that now the cells in here are single layer of cells, and these are secreting this, the cerebrospinal fluid, and they tend to be in the proximity of blood vessels. This is blood capillary, and then immediately there is a connection with the two, and maintaining the structure and ionic stability, and is circulated from the ventricles to the central spinal canal and the perivascular spaces. The problem with this is there is a blockage of the channels in which these flow. And that would be something that would be caused if the liquid would be way too dense with more frequency or more easily. Then there is a problem that the brain tends to increase in size. They become hydrocephalus. If this happens when the skull is still not completely closed, this is a problem that could happen soon after birth in some of the cases. And these are I don't know whether you guys can see easily the arrows. If I have designed myself this slide, I would have probably used a different color for the arrows. But you can actually follow the arrows indicating you the way this is flowing within the central nervous system. And then these particular locations and where the choroidal plexus is primarily located, and how they contribute to the overall circulation of this fluid. So everything is covered by the meninges for protection, which provide this layer of kind of spraying physical protection, and then it's bathed in this liquid that provides a second layer of protection. There is another view of the ventricles that are this is a lateral view and this is a frontal view. These are the channels through which the liquid is flowing, and of course they call them different names. The first ventricle, the fourth ventricle, the third ventricle, just call them ventricles, and that's for our purpose. When you guys go into anatomy, you will probably have to identify everything in every portion of these structures. But for us, at this point in time, the ventricles are particular structures within the central nervous system that provide the channels for the cerebrospinal fluid to circulate. So with this, we go into the peripheral nervous system. All right? This, again, is outside of the central nervous system. This is what communicates the rest of the world, if you want, the extrinsic environment and the intrinsic environment are going to be communicating with the central nervous system, letting them know what's going on and if there is anything to do about it. All right? So basically, we can define these as all nerves and supportive cells, glial cells and all those other kinds of things, Schwann cells and all that, which are outside the central nervous system. What qualifies your peripheral is to be outside of the central nervous system. We have been using these names from almost the very second slide that I used today. Remember that there are sensory neurons, sensory fibers, sensory nerves, and those are the ones that are actually receiving the signals from the environment, and they are sending these signals to the command post. All right, and then these are the afferent system nerve connections, and then the efferent or efferent, I remember the E as in exit, are the ones that go from the central nervous system to wherever they had to perform a function. And those are considered motor because this is a fundamental type that allows us to move our bones and move around and do our push-ups if we ever do them. And then there is a number of so-called ganglia, and there are some of them which are more important than others. And at the ganglia, I was talking during the break to one of you guys, and they asked me, so what are the ganglia? Okay, well, the ganglia, and the example I used was, I used the metro. You know, you go to ride metro downtown. Well, if you're willing to get in trouble and go to downtown and ride metro because probably it's closed, or you end up going on a bus or things like those. Don't tell that I made these political comments. What you find is that if you want to move from there are places, there are stations where there's only one line going on, okay? And you go on the red line, and you can go to my house almost. From this end, you don't have to change. But there are some places where the blue and the yellow and the red, you can move from one to the other. There are places that are nodes. There are connections. There are places where plenty of fibers, nerve fibers, come to go there. Some of them are going to go this way, some of them are going to go that way, some of the one is coming from here, from there. And there's this kind of node stations, which are pretty much called ganglia. And for the peripheral nervous system proper, one of the most important ones is the so-called dorsal ganglia. And for the autonomic system, there is a number of them that I'll show you in the picture, where there are the two portions, or the two branches of the autonomous system. The so-called sympathetic and parasympathetic branches are going to be getting together and communicating and form this set of ganglia along the different places. So in the peripheral nerves, basically what we find is this huge set of axons which are packed together in fibers, and the fibers are going to be becoming more and more complicated depending on the number of axons that they pack. And of course, if you look at each of them themselves, what we've been looking at is an axon which has been myelinated, and it could be going in one direction, it could be going in the opposite direction. We need to start looking at the coverings of this system that protects the entire wiring system, and is connective tissue. And depending on the location of the connective tissue, we call them the epineurium. There's more information on this a little later. The perineurium or the endoneurium. And that is fundamentally related to insulating and protecting the nerves from any damage. In terms of the afferent system, one of the important components is the, as I said, the dorsal root ganglia is located in the posterior part of the spinal nerves, and some of the cranial nerves will look into the origin of these things. And pretty much this, as I mentioned earlier also, most of the peripheral nervous systems are made with pseudo-unipolar neurons. The pericardiums are generally round. The range of sizes is incredible. Some of these nerves could go all the way from the hip all the way to the to my toes, and some of them are much shorter than that. So in terms of sizes of these packages of nerves, there's all kinds of differences. But the general structure is about the same, and in the majority of the cases they are pseudo-unipolar. Remember, those are the ones in which the body of the neuron is kind of in the middle of the whole thing, so is able to coordinate what's happening in that end and what's happening in the other end. All right? So this is precisely what I just tried to say. The processes from these go in the two opposite directions, and the impulse, and that's the other thing. The impulse could actually have nothing to do with the pericardium. The pericardium stays kind of out of trouble by being on the side, and the neurons are surrounded by the tissues of connective tissue that I just mentioned before. With regard to the efferent system, the cells are primarily multipolar, and there are different kinds of ganglia in there that are going to be identified depending on the function and the connections that they have. And we need to distinguish then different structures in the peripheral nerves, the axons, the tissue coverings, and how the myelin is distributed. This is what I kind of warned you already about, and we need to understand what these things are. These are the three layers of connective tissue that I mentioned earlier. There is one that is actually covering the entire nerve. That is going to be the epineurium. Then within that nerve, we have bunches of nerve fibers. And this one, each of them is going to be covered by a second layer deeper than the epineurium that is going to be is called the perineurium. So this portion here will be the perineurium, which is immediately underneath the epineurium. And then each of these little things, the little nerves in here, is surrounded by connective tissue that is outside of the structure of the nerve itself, and these are called the endoneurium. So there's three layers of connective tissue which are actually protecting each of these nerve fibers, these nerves going from one direction into the other all the way along. Yes. I'm sorry. Yes. Oh, before this one? This is not showing the autonomic ganglia yet. We are not dealing with the autonomic system yet. We'll deal with that towards the end of the class. Okay. So myelination. This is simply a repeat of what we talked about before. It's important to realize that the majority of the nerves in the peripheral nervous system are covered with Schwann cells forming this typical structure, and it makes more sense. Remember that I just said one nerve could go from my hip all the way to my toes, and if you want the transmission of the signals done quickly, and I'm not that tall, but if you are much taller than I am, then the transmission of the signal has to be accomplished as quickly as possible, and it's much more efficient to have this kind of a structure in which the signal jumps from here to here to here, and the transmission is, as we said before, 30 times faster than when you don't have this kind of structure. Now that doesn't mean that every single every single axon is myelinated, but this is the unmyelinated neurons. As I mentioned before, they also have Schwann cells around them, and the difference is that they do not wrap around them so many times. They do not form the myelin structure. They keep having protective roles, and look at these things here, that the Schwann cells, and this is the nucleus, this is a section of one of these non-myelinated nerves, you can see the axons, all these axons are actually protected by what seems to be one single swan cell that is kind of making holes to protect them. And in making those holes, there's these points of junction, or these points of that create these kind of little breaks in the membrane of the swan cell, and this is what is called the mesaxon structures. So there is swan cells protecting, but they do not form the myelin. The idea is that you don't think that every time that there is a swan cell, it always forms myelin. It's only when it wraps around many, many, many times and provides so many layers of insulation. All right? So in spite of all these protections and all this myelin and all these things, there are occasional accidents, and you actually end up having some nerves which are damaged. And to understand the process, we need to talk about something that is called chromatolysis. We're going to introduce the notion that, and in a way we talked about this before, all these cells except the neurons in the adult divide, okay, are able to divide. The neurons do not seem to be able to divide. Until not that long ago, this was a dogma. Now it looks like there might be a subset of neurons that may actually have the ability to divide, but I will not get into that today. And then we're going to be talking about the actual regeneration of the nerve fiber. And this is the whole process. So this is a normal structure of a myelinated neuron that has the perikaryon and all these nasal bodies and all these structures with the myelin. And in this case, it's a motor neuron because it actually ends in the muscle. And there is a problem, right? There's been damage in this nerve. So one of the first things, and this takes some time, if it actually works, takes some time. But within the first 2 weeks, what you can find is that there is substantial changes in the perikaryon. The nucleus, which is the most important component of any cell, and it actually kind of goes away from the problem, right? So remember that it used to be located in the center, in the perikaryon, now becomes what is called eccentric, all right? Different from normal. That's what the word eccentric is applied also to people. It's different from normal, all right? So it goes away from the norm of having the nucleus in the center. And as you also can see in the scheme, actually represents very nicely, almost all the nasal bodies are gone, all right? In the meantime, the main player at this point is the swan cells. The swan cells recognize that something has gone wrong with this particular neuron and start proliferating. They start dividing. And of course, as you create more cells in the same space, they have to go somewhere. And the main where they can go is towards the brake, also because the brake releases some cytokines or chemokines that are attracting these cells to go to the position of the brake. And what happens is that now they finally establish contact. So the gap that had been created by the problem is now filled with cells that are proliferating and cover this gap. Soon after that, or almost simultaneously at about the same time, the perikaryon recovers the normal structure, and the cell starts trying to work as if a normal neuron. And if the process is successful, then there is the cells form this column. The swan cells form this column that is kind of telling the axon from the top portion is okay to continue now growing. So the axon at this point in time grows in the direction of the where the connection was in the other end in the beginning. And if everything was fine, within a period of time that they estimate about 3 months, everything is regenerated. There is myelin, swan cells in this place, but you see the difference in terms of completing the myelination as it was is not always accomplished 100% all the time. But this cell that has been regenerated thanks to the proliferative capacity of the swan cells is able to assume pretty much most of the normal functions. Sometimes, of course, this doesn't happen, and then after a number of months, the process doesn't work, and they continue to be degeneration. The column of the swan cells doesn't make the contact, and there continues to be degeneration of these nerves. And if that's the case, there's also been, whoever did this slide, this print, It was very nice because it's also telling you what's going on at the other end. If an organ like the muscle is not properly innervated, it's not going to move. And what happens when you don't move too much, your muscles kind of diminish. So whoever did this, it was very an interesting artist, and I appreciate that. Made my life much easier. All right, so let's talk now about the so-called peripheral nerve endings, which is what happens at the other end. And we know that some of them actually go to the muscles and to tell us how to contract and other things, but many of them actually go to other organs and pretty much are there to be able to receive information on how things are on that particular end. And so there is different structures that have been created in different tissues, and these are simply examples of them. We'll look at some of them in more detail next Monday when we look at these in connection with the muscles. But these are specialized structures in which the different degrees of innervation, in different degrees of what I could call collateral tissue is made in there to provide a place where the reception of the signal is located. And some of those, the majority of them are actually generally complex structures, okay? And we'll look at some of those. And we'll look at some of those actually at the in the lab portion to show you actual pictures of how to look. The diagrams that I showed you before do not do too much justice to some of those things. So let's talk about the autonomic nervous system. And as I explained before, the use of the word autonomous or autonomic is actually not really correct, because that would say that it's working on its own, and it's not working on its own. It's actually work under the direction of the central nervous system. Different portions take care of the couple of branches. There are a number of ganglia that we will show you later. This is places where nerves from one direction and the other connect, transfer stations in the metro terminology. And then there's two main branches, the so-called sympathetic and parasympathetic branches. Just from the beginning, I'll tell you that these two branches do different things in different places, but that they in general tend to kind of oppose each other. If the sympathetic nerve of a specific type is activating certain process, the parasympathetic connection to that same place is doing an inhibitory process, and vice versa. If the parasympathetic is inhibitory, the sympathetic are the other way around. I don't know whether I'm repeating exactly the same thing I did with the phrase in the opposite orientation. So let's look a little bit more in detail, and here is where the ganglia are going to start showing up. All right. So I hope that you can see that there is two different colors in here, the blue ones and the red ones, or pinkish one, and the full lines and the dotted lines. Okay, these actually correspond to different things. And what we have here is a portion of the central nervous system that includes the portion of the brain stem. Remember from the first slides of the second class, the midbrain and the medulla is kind of that thing that comes out of the brain and connects with the spinal cord, which is this one. And then there are different places that receive different names in the spinal cord according to the vertebrae in which it is packed. So we distinguish in the spinal cord for this purpose cervical, thoracic, lumbar, and sacral regions. All right? The sympathetic nerves all derive and they all connect with the spinal cord at the level of the thoracic lumbar connection. All these are the efferent nerves. These are going to be sending signals to the outside once they receive signals from the outside. And the outside in this particular case is anything that connects with the outside, like the skin, like the eyes, like the ear, and the sense of tact and things like those, or the inside, which are different organs. All right? So the efferent, in both cases, the sympathetic is presented in red, the parasympathetic is presented in blue. But the efferent is the solid lines, all right? The efferent in the sympathetic, it all comes from the spinal cord. The efferent in the parasympathetic system all comes from either the midbrain, the medulla, or the sacral portion of the spinal cord. So this is why the system is called craniosacral system, and this is a thoracic lumbar system. And basically what you find, and there is a number of ganglia, the sympathetic chain of ganglia, which are located in here, but there are different connections between them and maintain the overall structure, and also make connections with some of the some of the afferent nerves that come from the different organs that they innervate. There is different specialization many times because of proximity to the type of organs that they innervate, but that's fundamentally the way they work. I think I've gone ahead of myself because what I've been trying to say can be probably said with words, and then you guys can read it in different slides. The image is pretty much the same, and the notion is that this is a definition for those of you who wanted to know what ganglia are and don't like metro. Ganglia are groups of nerve cell bodies located outside. So it's not just the entire anything. It's just the cell bodies which are forming the fundamental ganglia. All right? I think I said this already, so we're going to go ahead. Oops. And here, what I wanted to identify is the name of the ganglia in the autonomous system, and we talk about three different ones. The paravertebral, para usually means something closer to. So these guys are very close to the spinal cord, and this is all a complete set of ganglia that are, oops, I think I'm going crazy in here. This is the paraventral ganglia. Then another step is the paravertebral ganglia, which are, again, important nodes where plenty of bodies and kind of halfway, let's say, between the transmission of the signal one way or the other. And then there is a terminal ganglia, which are located in the organs or the tissues where the signal is being received from. Okay, these are either superficially located or into the organ itself, but they pretty much are recovering the information on what's going on in there. So these are the three main types of ganglia in this system. This is simply telling you more details about these containing each of these types of ganglia. But as I said, the main function of these is the relay of the information from one group to the other. And the terminal ganglia are primarily in the parasympathetic one, and the paravertebral are sympathetic, and in the paravertebral ganglia, prevertebral ganglia is you can actually find the ends of one and the beginnings of the others. Same things, and this is simply to illustrate or to remind me to say something I already said, which is the opposing functions that are detectable between the parasympathetic and the sympathetic. Okay, so I put here an example of the neuroepinephrine. There's an O missing in there, by the way. Sorry for the misspell. There is a neuroepinephrine is released by the sympathetic nerves, and it actually works on the heart. Okay, the heart rate is increased by the neuroepinephrine. However, the acetylcholine secreted by the parasympathetic component, it actually reduces the speed of rate. So this is an example that gives you the idea of these two different functions of the two branches of the sympathetic system. And this is, again, another illustration of the two types of cells that I just mentioned. The heart increases, and this is because of the different kinds of cells and the different kind of connections between one type or the other, and the nature of the neurotransmitter that is released. So it's only the connections that make that possible. So this is a picture of a ganglia. This is the metro station, right? This is what you find. Is in the ganglia is see all these bodies of the neuron. Okay, so this is typical region in which you find primarily the bodies of the nerves. There's a number of nerves that converge in that particular place, and you were to do sections in different positions, you will find these kind of structures, typically with the nucleus located in a central position. There is these are the many cells, Schwann cells, I'm sorry, the capsule cells. This is now the fibrous covering of the entire nerve, and these are some of the ganglion cells that contain the nucleus in a central located. So this is typical, this famous dorsal root ganglia that is located very close to the actual spinal cord, and this would be one of the sympathetic ganglions that you find on the way to the transmission of the signal for the efferent system. And with this, I thank you for your attention. I don't know whether there are any questions now. By the way, talking about questions, since I'm a professor at Georgetown and I'm slightly far away, I don't spend too much time in this place, even though I like it so far. But I cannot tell you my office hours are from here to there because I will not be here. However, I recommend that you guys go to the first slide, probably the most important one. It has my phone, except for my bank account. All my information is there. So I usually am pretty good and humble, as you can recognize, about answering the emails. So if anyone has any questions, the students usually get worried about exams. I don't know that. I don't know why that, but anyway, if there are any questions that you cannot sleep because of problem with understanding how the epineurium goes around, email me. But if you do, or call me, the phone numbers are there, and 2 email addresses. I mean, if you don't contact me because you don't want, but if you contact me by email, somehow identify yourself as someone from GMU, because I am also very efficient at deleting with the delete key. I use it frequently when I don't recognize anyone has sending me anything. I'm wonderful at using the delete key. Okay, so I'm at your disposal in terms of answering any questions now or whenever they come up. Should we take a break and then we get into the lab? There's about a number of images that we want to show you so that you guys recognize in reality what I've been talking about. So let's take another 5 minutes and then get back and we'll finish soon. Thank you. Are you typing in something?
Yeah, it's just that. We have to
Cop-
make sure if this.
Actually, we haven't talked about copyrights yet. All right, guys, let's go and look at some of the real pictures of the real thing, and then we are all going to have an easier time, and maybe we can even eat something at some point. Let's look at histology here. All right, so this is the cerebral cortex of someone who's like this, okay? Simply in that position to fit it in the slide better. You can see is the molecular layer and the portion where the most of the bodies of the large bodies of the cells is. This would be the pyramidal cells, and this would be the pyramidal segment. But this, again, as I said, this is the way this will look will again depend on the type of staining that you have. So this will be the molecular layer, and then will become more cellularized. And the way this doesn't stain that much is because the axon did not get stained. Questions? All right. Now this is the cerebellum. This is the different layers. Remember, in the cerebellum, the gray matter is outside, which doesn't look very gray in this particular stain. And this is what I've been saying about the influence of the staining. And then this is the granular layer, and much more smaller multipolar neurons
form this very dense, cellular dense area, compared to the density of these other ones where there is more axons in all these location. And the white matter is in the center again, and the gray matter is in the outside, right? And these are the same kind of layers that we've been talking about in the lecture. Questions? These you probably can recognize. What by the way, there's a misspell here too. I think this is a J, Purkinje with a J, not with a G. I apologize dearly for such a terrible mistake. Sorry. But what is important here is to realize is the granular portion of the multiple cells and the Purkinje cells, which are very peculiar. This pear-like structure of the neuronal body and the huge ramifications that come out of the body of the Purkinje cells. They're larger than many other cells, and there is definitely defining the structure in which they are located. Any questions? In case you haven't seen enough branching for a neuron, there is a Purkinje cell, again with the same spell, but this is what people talk about in some places. They talk about arborization because it looks more like a tree than anything else. And this is an individual neuron. Imagine a whole layer of these, the possibilities of connectivity between different kinds of cells that this provides. As I said, these cells have important roles to play in the past. Now, who thinks that the gray matter is inside and the white matter is outside? Huh? You think so? Well, whoever did the stain fooled us completely, because it is actually the other way around. This is supposed to be the white matter, and this is supposed to be the gray matter. So when you look at histology slides and things like those, one of the things that you try to identify is how was the stain performed. Okay? Because otherwise, if you are not familiar with the structure, you're going to be having some problems. But despite of the overall problem with the stain, is the peculiarities of this particular stain, I guess structurally speaking, we see what we needed to see. In the spinal cord, we have two very clear regions that are stained very differently, depending on what's located in them, whether they have neurons and nerves or neurons and glia and nerve fibers and glial cells, and the famous H with the dorsal and ventral horns, anterior, posterior horns, and the H being closed by the central canal. All right? Questions? Now you want to distinguish something that is not even white or black or gray. This is another different stain, and in this case it's something called Luxol Fast Blue, and amazingly nothing looks blue. You see orange and green. These pathology people really try to fool us all the time, but you can see the same structure, the famous H. Okay, the gray matter is located in the H, the central canal, the white matter, which is cellularly and structurally different from the gray matter in the outside of the spinal cord. Questions? This is in the spinal cord, and what it's trying to illustrate is a point of connection between the white matter and the gray matter. And if you remember, we said the gray matter contains cell bodies, which you can see, plenty of them, and the white matter contains primarily, both of them contain glial cells, and probably something like this tiny nucleus in there is coming from glial cells, and this other one is the white that contains primarily fibers, nerve fibers, and primarily axons that is stained in a completely different way, and you see the transition from one to the other. Questions? This is a magnified version of one of the cells in the cerebral cortex. This large, in the pyramidal layer of the cerebral cortex, and what you find in here is the huge body of the cell. You can actually see plenty of the filaments inside the cell that stain, and the nucleus centrally located with a very prominent nucleolus in there, which actually is a very good characteristic of the majority of the neurons. Another neuron seen in up close, this is the pericardium. The nucleus, the staining in here doesn't allow the staining of the nucleolus as a single individual entity. It gives you a diffuse staining of the nucleus. But you can see if you get close to these slides, also the extension of the neurofilaments that go actually into the dendrite, which is this portion of the cell. Any questions? This is a ganglion. As you can see, it resembles very much one of the diagrams that I showed you at the end of the second lecture earlier today. You can see all these protective neurons, all these protective cells. Some of them will be the Schwann cells with the proper, indicating the nuclei, and then you have now some of these nerve cells in which they have the proper characteristic. With some of them in here might have the myelin, and you can see the Schwann cell kind of folding on top of this one with the location of the nucleus in a centric to eccentric position. Questions? All right. So these are now taken from a sensory neurons. What you see here are the neurons are, what we're looking is at the nuclei of the sensory nuclear neurons. And then these cells are trying to illustrate something called satellite cells, which are also providing a layer of protection in the formation of these structures of the sympathetic nerves. So the satellites are these small ones, and the nucleus of the actual nerve cells are located in here. Questions? This is one of those specialized structures that I mentioned earlier that is included in the category of nerve endings. This is how the nerve goes into the particular organ. This slide is taken from the GI tract, okay? And this structure, Dr. Auerbach determined that this had to have his name. So this is the so-called Auerbach's plexus, and you can find this is the nerve ending. You can see this is, of course, remember who goes into the organs. It's the parasympathetic portion of the autonomous system. So these are in the GI tract, and therefore these are parasympathetic neurons that are coming to this point, and they're going to relay information on what's going on in the GI tract to the central nervous system. Most likely, and this I'm saying by memory, but because of the location, the ganglion that is associated with these had to do with the sacral portion of the spinal cord. Oh, I'm sorry. Any questions? All right. This is just one single myelinated axon. And it shows you that the myelin, actually the inside, the myelin is the dark stained portion on both sides of it. And the inside that looks as pale as the outside, probably because of the staining being directed just to the myelin portion of it, is what would be the axoplasm. In other words, the cytoplasm of the axons. And the break, the discontinuity here is what is called the node of Ranvier. There is no myelin in there. And because this is a stain directed to the myelin, therefore this portion here doesn't stain. Okay? Yes, sorry. In the myelinated axons, remember that there are Schwann cells that kind of wrap around, and there is a number of them, and in between them there is a space. And that space is the discontinuity which is shown in here. In other words, what this is showing is the axon goes in the inside, and this is a myelinated, a Schwann cell that is kind of wrapping around, and then ends it here. There is another rhombier, and then another Schwann cell is wrapping this portion of the axon. Am I explaining myself? If it is an axon, and if it is some axon that's myelinated, I bet you half of my salary, which is not that much, that it will be another rhombier anyway. I'm not going to bet the whole thing. Come on, guys. All right. So, did I answer your question? Thank you. Any other questions? Now, if you cut the nerves in a transversal form and you stain for myelin with something that it definitely stains much darker than in the previous slide, this is what you see. Okay. You see plenty of nerve cells which are surrounded by myelin. Okay. There is also some portions of connective tissue that you can see here, standing kind of brownish in between them. And there are, as far as I know, the guy who did this slide has been very good at showing myelinated portions. And we cannot deny that maybe somewhere in between there is another rhombier that, because it didn't stain, we cannot see. All right? But this is a transversal section of many myelinated neurons, and that is where you can see in there. The inside of each of those little circles is the axoplasm, is the inside, the axon is in there. Questions? Now this one is a composite of two different things. One is in the left side, there is the same picture as before. The neurons have been cut transversally, and therefore you see the layers of myelin that are surrounding the axons. In this case, we've been lucky enough that is also an unmyelinated axon that has been captured in the image, and you see a significant difference between this one in here and the other ones in there. Okay, this one you can see the Schwann cell that is actually the body of the Schwann cell is showing in addition to all the layers of the cellular structure that surrounded the axons to form this particular structure. On the right side of the image, what you have is a longitudinal section of nerves. And then what you find is a number of nerves going together. All of them, in all these cases, you see that there is a myelin on both sides. Okay? And in between the axons and is the space in here, for instance, or there will be the space in here. Okay? This is a connective tissue that is maintaining the integrity of the whole bundle, that is going to be involved in transmission of signals in one direction or the other. The one that surrounds the axons, if you remember, there is the epineurium that surrounds the entire nerve fiber. The perineurium. The epineurium that is surrounding bundles of nerves, and then the endoneurium, which is the one that is surrounding, is a connective tissue surrounding each of the axons themselves, and this is what these layers of connective tissue will be corresponding to, the endoneurium. Questions? Yes? This one, yes. Yes. The nucleus doesn't stain properly with this stain. Now we go to astrocytes. Remember, these are the cells that look more like starfish, and there were different types. They were the protoplasmic astrocytes, which I think I said a couple of times that they are typically located in the gray matter. I'm going to show you very soon a picture of the fibrous astrocytes, and I want you to realize that these in general have a good number of processes, but they tend to be shorter and a little fatter or wider than the astrocyte, the fibrous astrocytes. All right, so these are in the gray matter and the central nervous system, and in the spinal cord also. And this is a huge difference you can see with the fibrous astrocyte. There's many more processes and they're much more slender. And if the other ones were in the gray matter, these are frequently, not frequently, only located in the white matter. Okay. This is the oligodendroglia. All right, oligodendroglia or glia. Oligo means, again, scientists like that. Oligo means a small or few. Okay, and the name of these guys means that it has little number of processes. In fact, the other half of my salary I will bet with anyone who can see anything that resembles the processes of the astrocytes we've seen before. There's kind of bumps outside, tiny little bumps outside that are kind of not even elegant, but it has an important function in terms of protecting and actually being able to be located in places where the other larger cells cannot be placed. Questions? Yes. Here. Could be anything. There might be a mixture of connective tissue, it might be a mixture of neurons, because this stain might be specific for glial cells. There's a number of proteins which are, of course, like in these cells, which are present only in one type of cells. There's a protein called GAP43, for instance, which is present in glial cells, and you cannot find in neurons, for instance. So I don't know whether this staining is directed to a particular component. So it could be connective tissue, it could be neurons, it could be anything. But the message of this slide is to show you that these are smaller glial cells, and they have essentially almost no process. Yes? I'm sorry, I cannot. Oligodendrocytes, yes, you can actually use it that way. Yes? They are located pretty much everywhere. Okay. Almost any space within the central nervous system that is not taken up by anything, the glial cells are going to come and take it up. Okay. They are also mobile, these cells. They can actually migrate slowly, but they can actually migrate, so they can fill any spaces. If there is any damage, let's say because of an accident to some of the meninges, and there is a gap in there, immediately it's going to be filled up by. In a way, although this is a kind of a far way of comparing, the majority of the glial cells are, for what other tissues are, the fibroblasts. Fibroblasts are very mobile. Epithelial cells are not. Neurons are not mobile, and some of the other components may be, depending on the proper stimulation. Microglial cells, the ones that behave like macrophages, these can actually be in a way like, you remember, you probably are too young, the Pac-Man kind of game, these guys eating all that stuff, they could actually phagocyte almost anything, and for that sometimes movement is required within the space that you have, of course. Anyway, any more questions about these little fellows? No. Okay, so now this is a better picture compared to the one I shown you in the diagram before. This is the epi and peri and endoneurium. Okay, these are the groups of nerve fibers that are actually forming a huge fiber itself, that then you go to internal fibers, and this is the external portion of the whole thing that it should be wrapping around and getting out of the slide. And this is the layer of connective tissue forming the epineurium. Epi means the most external part in there. Then each of these fibers that's formed by many neural cells is surrounded itself by a second layer of protection called the perineurium. And if we were to go inside and actually magnify this, but we cannot do it with this system, you will see that every single axon is surrounded by connective tissue, which is outside of the myelin layer, if there is myelin layer, and that will be the endoneurium. Okay, so 3 layers of packaging. You'll see that this is something that happens in nature. When we look at the structure of the muscle, we will also see The name will be different, but there will be an epi and peri and an endo. Okay? Questions? Yes. This is found in the peripheral, well, yeah, the peripheral nervous system, or any fiber that goes into the central nervous system. Okay, remember that they go at some point, they get connected, right? But inside our brain, you don't find these kind of structures. This is the nerves. This is more questions. All right. This is another. You will see some more of this next time. Next Monday. Remember the specialized nerve endings that I've been talking about earlier. There are different organs have different structures. The structures tends to be complex, and this is what is called a muscle spindle. It's called a spindle because if you look at this in regular muscle, it really looks like a spindle shape. This is simply a transversal cut of that spindle. And is a point in which inside the muscle there is a point of connections of blood vessels and nerve and nerves that come and go out, and they all coalesce in that particular location, and this is a structure of it. So you can actually see here, these are all, start getting used to see these things because I'm bringing some of these things on Monday too. This is muscles cut transversally, muscle fibers, and then this structure is the one that actually connects the nervous system and with the blood vessels and in a specialized form. There is, of course, the synapses that go to the muscle that are completely different than nerve endings, but this is very specialized structure. Questions? All right. Another very specialized structure that looks much better here in that diagram that I showed you earlier. This is the Pacinian corpuscle. This is in the skin, and this is pretty much what allows us the sensitivity to touch, pressure, and vibrations. All right. There is lots of layers of connective tissue surrounding the nerve tissue that is going to be pretty much detecting the signal. This is, nature again is very clever, and you can call it nature whatever you want. But if you have nerve endings very close to the surface, the outside surface of the cell, you really want to make sure that these guys are insulated and properly protected. Okay, so never enough layers of protection is enough. All right. Questions? So this is not a myelinated neuron, okay? Just in case anyone thinks that would be the case. And anyone will say, why not? First hint, why this is not a myelinated neuron. You see more than one nucleus around the center, which is the nervous cell? All right. So what this means is that kind of fibroblast-like looking thing is sticking around, and each of them has its own nucleus. If it was a section of a myelinated neuron, as I was pointed out before, you can see actually the body of the Schwann cell that will be wrapping around in a cell of this kind of structure. Questions? All right. This is also close to the skin. Actually, in the skin, in the dermis of our skin, is Dr. Meissner gave it the name to this one, and these are kind of finger-like protrusions into the epidermis and the dermis and the epidermis of the skin that have the same sensitivity. We have plenty of these things in our fingertips and in the sole of our feet, okay, and they are responsive to light touch, and what's interesting about these ones is that they are formed by unmyelinated nerve terminals, and these are surrounded. Typically, the protection is given by connective tissue that's formed at these structures, and frequently located near blood vessels. And this cap here is an indication of the capillary ends in the same location. So these are the different layers of the skin, and these things, finger structures that go into it. Questions? Now this is a longitudinal section of a nervous tissue. And I want to illustrate here, because of the question before about the interruptions or the breaks, how an interruption in the myelin and the nodal ranvier will be located. You can see the Schwann cells. In this one, you can actually see the nucleus of the Schwann cell, which is located next to the axon. And the myelin is surrounding the axon. And here is a nodal ranvier. There is another nodal ranvier. There is another ranvier in here. So this is a way in which the stain has done multiple cells at the same time. The one we saw a number of slides ago that related to, brought the question about the interruption between the two portions of the single cell that was myelinated. This showed you the general picture. All these nuclei correspond to the Schwann cells, and then the nodal ranvier are the breaks in the myelin sheath and where the depolarization events take place. Questions? Now we're going to look at some electron micrographs. Okay, this is looking at the cell body of the perikaryon. This is the of one cell in a tissue, and I try to highlight the numbers because in some of the original pictures they didn't do a good job. Of course, you can immediately recognize this as the nucleus. These things, which are labeled number 2, are mitochondria with a little bit of blood. I don't know, from here I can see, but you could actually even see the cristae, the internal membrane of the mitochondria in there. The endoplasmic reticulum, which is a great number of membranes, and this is a rough endoplasmic reticulum, so these little things are most likely ribosomes. Number 4, these kind of light vesicles that are around in this area next to another ones that look more like the endoplasmic reticulum are the Golgi apparatus. And there is a number of, in the neighborhood, there's a number of myelinated ends, okay? And in this particular position, it's small, but it still could be visible. This cell has actually a number of vesicles inside, and is getting close to the membrane of this other cell. And you can see here another one. You can find presynaptic vesicles, so this means that this is a postsynaptic cell, and these are presynaptic cells that are coming to it. Questions? This is to give you a better perspective of the endoplasmic reticulum structure and how this is really the rough endoplasmic reticulum. The Golgi apparatus, here they call it complex, but it's actually Golgi apparatus. This is a fundamental structure, probably you guys know about that, in terms of the creation of secretory vesicles that are going to be channeled through the microtubule system to where they have to go. Of course, in a neuron, this is supposed to go down the axon. The nucleus, the endoplasmic reticulum, and the mitochondria are the number 2, which are right here. And sometimes the problem with the electron micrographs is that if they are not properly exposed, I don't know, you cannot see too much detail. Anyone can guess the kind of cell that we have in here with this heavy stain thing? Could it be myelin? Dark staining structures? Very thick, the structures in there. So the axoplasm, these actually, the arrows point to the inside of the cell. They are not pointing to the outside, okay? So this is simply the inside of the axon, and there is the axoplasm, all right? There is mitochondria in there. There is a number of filaments. There is actually a neurofilament here that have been caught, and it's slightly thicker than the other filaments inside the cell. The myelin is this one here, of course, and this is the next cell next door to it is a non-myelinated cell. It's surrounded by surrounded by a Schwann cell but it's wrapping it just once. And in the process of wrapping it, is actually forming a gap when one end of the cell gets to the other one, and that forms the so-called mesaxon. Remember that there was a slide that we showed you in the class that when one swan cell wraps only once, the two ends of that wrapping action are going to get to a point that they touch each other and they form this kind of gap. It's called mesaxon. Anyone would have any suggestion of what this may be? What do you think is in there? Vesicle. So what could it be related to, right? Could this be the portion of a presynaptic cell? Okay, good. Questions? All right. So this is, again, another synapse. It's called axodendritic because there is an axon that is the one, obviously, that has the vesicles in there. This is the presynaptic portion. There is the portion in which this one is joined to this other one that's supposed to be the dendrite. And then there is the synaptic cleft and the particular structure that is formed between the two. All right? So this is a postsynaptic dendrite. This is the presynaptic axon. And then you can see typical structures that were seen before, like mitochondria and things like those. And some of the cells which are surrounding these are myelinated cells. These are astrocytes that are wrapping it only once. There is another one here with different kind of much more, looking much more like the structure of the synaptic structure that we've seen before. There is, again, the structure that is going to be delivering all this physical content into the dendrite of an adjacent neuron to keep transmitting the signal. Excuse me. Any questions? It's an oligodendrocyte wrapping the, of course, it's wrapping something else, but this is the overall structure of the oligodendrocyte. This is the nucleus. There is the different typical regions of any well-behaved cell. There is endoplasmic reticulum. There is a Golgi apparatus. There is a number of myelinated axons in here, and a number of 5 here, right there. Although the picture doesn't do too much justice, and the positioning of the number, I tried to change it, but it came to me. By the way, I had to, you guys realize that I inherited these things. This is not what I do on a daily basis. I don't look at these kinds of things. And I'm used to move things around. You look at signal transaction maps and things like those, you move your signaling molecules in your atoms around. But this is something that actually these slides, with all respect, come from Dr. Peter Andrews, the person who used to teach these. So I received these slides, and I was surprised that I cannot move. And the only thing that I could actually do is change some minor things, but I could not move around. But this thing here, I'm going to try to delineate it for you. This thing that goes, and then it goes under the number. This is a non-myelinated axon. Okay. You can tell easily which is myelinated and which one is non-myelinated. Okay. Whether you can see that it's an axon or not, that's a different story. Questions? This is an astrocyte. Basically, is an astrocyte that is going to be wrapping something. That's what astrocytes tend to do. Is basically what this one is wrapping is a blood vessel, right? So this is the lumen of the blood vessel. Of course, you guys know that the blood vessels are formed by endothelial cells, and that's something that by now you have studied, or you haven't studied that yet. You haven't studied that yet, so you probably know more about that than I do. And this is the endothelial cell, and so this is the nucleus of the endothelial cell. There is some interesting structures here that are so-called tight junction. Again, the endothelial cells is wrapping and creating the capillary, And at the opposite end, what would be the mesaxon for a Schwann cell wrapping something, this structure in endothelial cells form a tight junction. Okay. There is, of course, the basement membrane, there's the pericyte, and then the astrocyte processes are wrapping around it. Okay, and it's these 6 that goes all around, and you find again on the other end, surrounding the capillary, there is an astrocyte that adds an additional layer of protection. This is one of those places in the peripheral nervous system that is arriving into the skin, and they are arranged into the so-called fascicules, and is simply indicating to you that there are certain myelinated axons. There is some unmyelinated axons, very clearly distinguishable here. These ones that do not have this thick structure do not have myelin, okay? And then everything else is connective tissue. And this connective tissue, because of the location of what it is the endoneurium, okay? So that easily, very easy, this slide makes it very easy to show what the endoneurium does. It's maintaining, it's kind of the quote-unquote glue that maintains all these myelinated or non-myelinated axons together. Yes? I'm sorry? These are axons. This one? This is the myelin. This is the inside of the axon, and these portions are different sections in which you have cut the Schwann cells around it that are actually the bodies of the Schwann cells. Any more questions? Yes. I'm sorry, but I cannot hear one. Oh, the Schwann cell is much larger than the satellite cell. It's always located only in one position relative to the axon, and the satellite cells may be many surrounding the axon and are much smaller and all nucleated. So you're going to be able to distinguish it that way. Any more questions? Well, it's been great working with you. Have a great weekend. I'll be back on Monday, and we'll discuss the muscles.